Okay. Well, welcome everyone. Welcome, Lexi. Thanks for joining our work in progress seminar this week. This week we have Lexi White, who's going to talk about Project Itch in Rhode Island and share some preliminary results and insights with us. So just for an introduction, Lexi is a research associate at URI's Center for Vector-Borne Disease, and she's a disease ecologist specializing in ticks and tick-borne disease systems. She completed her bachelor's in wildlife biology at Unity College in Maine, her PhD in ecological sciences, and a graduate certificate in modeling and simulation at Old Dominion University in Virginia. And Lexi has research experience in surveillance, educational outreach, spatial ecology, and mathematical modeling. Very impressive. Welcome, Lexi.
Hi, thank you for giving me this opportunity to talk. I forget that you do an introduction, so I totally have a slide that's about me, but we will skip that part. Most of my travels
All
have been
right,
up
go
and down
for
the
it.
No, it's the same, I promise. But most of my travels have been up and down the East Coast, but my random bag of jobs along the way has given me this really cool skill set. And so I have this toolbox full of several things, including a lot of tick surveillance in different areas, including invasive species, and a lot of work on Lone Star ticks, as well as educational outreach methods, math ecology. I have a lot of like wildlife background. I've helped with different deer check stations or deer calls along the way. And then with URI, most recently over the past year, I have learned a lot about tick colony maintenance, and everything that goes into that. So that's a bit of foreshadowing because at the end, I'll wrap up with talking a little bit about the tick colony. But for now, I'm going to talk about Project Itch. And so Project Itch overall, right? I'm talking, I think, most we to new VEC folks. So we know what Project Itch is because we talk about it a lot. But across new VEC partners, we're looking at is tick control helping? And each area of New England kind of broke this up into different ways. With the overarching question, is tick control helping, but with different nuances per state or per region. Um, this has provided a really great opportunity as well for internships and mentoring in that sector. And so we've corrupted a few more students into the tick ecology world through this program, and it's been really fun to work with them the past couple of summers. Um, but with Project Itch and if tick, well, is tick control helping? There's so many questions surrounding this, and a lot of what I hear from homeowners is, should we spray or not? What do we spray? When do we spray? How do we spray, right? And the layers of this are quite complicated to get it consistent across scale. And there have been several studies, many of which the audience has completed, right? To try and regulate this question. But even with all of this variation, are we able to see a signal in real time people's backyards, given the treatment that they are just getting on their own, is how we kind of angled it at URI. And so these are just some of the companies that folks have access to. My personal favorite name, I'm just gonna say that, so there's not too much bias here, is Tick and Skeeter Deleter. But they have all these clever names and clever slogans to try and hook you in, right? At the end of the day, they are companies to make money, right? They are pest control organizations or products that you can buy to apply within your own backyard. But navigating what is best and how you define best as a homeowner is a difficult question. And a lot of this can depend on the ticks in the area. And here in Rhode Island, we have 4 main species of ticks that you'll find in your backyard. There are others. But predominantly, our story has always been about the black-legged ticks and the American dog tick. Most recently, Lone Star ticks have started moving into the area, right, coming in from the east, and their prevalence has been increasing. And then just in the couple of years that I've been here at URI, we have been able to see the longhorn tick increase drastically in numbers as well. For our corner of Project Itch, these are our surveillance sites. It comes out to be about 175 over the years. From 2023 to 2025, we've sampled in May and July. We try to aim for that peak black-legged tick season, our black-legged nymph season. 91 of these properties have been treated, 84 are untreated. And the median property size has been about that 1.8 acres. Yes, we have some larger properties or some smaller properties, but really aiming for that ideal tick backyard, right? And to navigate so we can compare across. But here on the map, you'll see kind of a Halloween palette of colors with orange and gray being untreated and orange being treated properties. And so spatially, we tried to get diversity between treated and untreated, so that way we could control for spatial differences in tick populations as well. In terms of methods, we visited each property twice. While we were at the habitat or at the backyard, we would sample along habitat zones A, which is the direct edge of the backyard where it meets the forest. And then zone B. Zone B is just inside the forest, up to about 3 meters. And so we would do 10 30-second flags in that each of the zones. We would ID the ticks in the field. So each 30-second interval, we would stop and write down what ticks we collected, and we would ID them then, and then go back into the lab at the end of the day and confirm these IDs, which was fun for me to see what the students had learned over the summer and to see if we found any different or species that we weren't expecting. So like this summer in particular, we found a lot more longhorn ticks than I think we were anticipating. After this, the ticks get sent off to Dr. Zhu's lab for molecular ID and pathogen testing. And then from the results that we get back there, we send out these personalized letters to the homeowners. And so our initial year in 2023, we sent out a letter to the homeowners, and it covered how many ticks per hour. So whatever number of ticks we collected from our 2 sampling sections or sessions, we multiplied by 3 to get this ticks by hour number for the homeowners. And then reported each species and how many we collected. It also included the infections status of the black-legged nymphs that were collected as well, and let them know that they were more than welcome to contact us with anything or any questions that they had. Through these results letters, we got a lot of feedback. Folks wanted to know, okay, you found 10 ticks. Is that bad? Like, is 100 ticks bad in Rhode Island? What does your average backyard look like? And what is typical? So for the following year in 2024, and we hope to do this again this year, we presented the homeowners with a chart, and they were very receptive to this. And so we marked the average number of ticks that were collected in untreated and treated habitat. I'm like not using my laser, sorry. In untreated and treated habitat here. And so that way they were able to compare, okay, you found this many ticks in my yard. How does that compare for other untreated yards? How does that compare for other treated yards? And through Project Scratch, I was actually able to deliver some of these results to the homeowners in person and have this discussion with them. And it was really enlightening to me because if we're talking about is tick control helping, how do we define help, right? And so if I still find 10 ticks, does that mean that my treatment's effective or not? And so it allowed for these conversations about these gray areas when it comes to tick control. One of the other things that we included in the letter is how many ticks collected in zone A as well as in zone B. Depending on the application method for tick control, one would expect that in zone A would have the least amount or the fewest amount of ticks, right? And then in zone B would have more or hopefully less, right, than farther in the woods. But overall, zone A, that immediate edge of the forest, if I'm doing tick control, I want to know that I'm protected in that area. And having that insight into for the homeowners really allowed them to establish some sort of risk of, okay, I'm in my backyard versus I'm going to dump leaves into the woods, and maybe change their behavior accordingly. So a lot of what we've been talking about is tick control helping, and how do we get at answering this? At the broadest level, I guess we want to know that treated yards have fewer ticks, right? If treated yards have fewer ticks, then tick control is helping. And so that's our first question. Our second question is looking at the types of treatment. So the pyrethroids versus others, like the minimal risk natural products. And then also, are we able to see these differences for black-legged nymphs in terms of what control is working, or are we able to see it for all tick species, especially where we're in an area with Lone Star ticks and longhorn ticks, where there aren't as many control studies on them, or the control studies that have been done on Lone Star ticks have reflected that they're a little hardier um, and harder to knock down than the black-legged ticks. So starting to break down the data. So the students this summer were very, very engaged. We had Val, Emma, and Mason, and they wanted to know, right? Like they wanted the answer. And so they worked with me to enter the data. And so they really took this on and entered the 2025 data. And so for Mason's end of the year presentation for a summer internship, he presented the next couple of graphs. And I think this is just an interesting look at the higher view of our data so far. So in 2023 to 2025, the number of properties we sampled didn't change very much. I will say the first year was 57, the second year was 56, the last year was 62. So yes, there were a couple more properties, but this seems to be quite a bit of an increase overall for 2025. And then again, just looking at 2023 and 2024. Oh, let me go back real quick. This is for untreated and treated properties combined. So just for our study overall. If we break apart untreated and treated, so treated here in red, untreated in blue, these differences look good, right? If I were to ask you, is tick control helping? Yes, treated yards have less ticks than untreated yards. However, when we entered the data, when they entered the data this summer, right? Mason showed me the graph and I'm like, are you sure? And we went back through and this is the data that we got this past summer. So we're noticing less of a stark difference, right? And there could be a lot of factors that are involved with this. We collected a lot more longhorn ticks and Lone Star ticks this summer, as well as what type of treatment, right? Treatment could be anything from tick tubes to Wondercide sprays in backyards, right? And we don't know the exact timing of treatment. There are a lot of variables that go into this. So again, just looking at this question, using a bar chart of the average number of ticks collected and treated, and then the average number of ticks collected and untreated for 23 and 24. I believe Tom's presented these graphs before to this audience, and you can see the standard deviation. Here, just again, looking at it, we're getting a bit of a signal that untreated yards do have more ticks than treated yards, just for 23 and 24. But once we add in 25, our signal is lessened. So we are noticing that something happened in 25 that we really need to dig into more. Again, another way of looking at the data. So box and whiskers plot where the thick line here is the median of our data. And then you have the lower and upper quartile for the data. And then you can also see outliers here for some of the sites that may have been skewing our data. So 23 versus 24, treated number of ticks, untreated number of ticks. If we add in this past year's data, all of a sudden, again, our signal kind of this not stark difference, but this difference that we're noticing here between treated and untreated is much lessened because of something that's going on. So one of the ways that we started looking at this most recently is just by pulling apart what types of treatment are we talking about. And so if we look at the minimal risk natural products, they're in green here, red being the pyrethroids, blue being other, gray being unknown due to like discrepancies in what the homeowner told us, which is a whole other facet of human study research, right? That I'm still digging apart. So hopefully I'll have more answers soon. But it's really helpful in this particular scenario because you can see our outlier way up here above 100. This particular person treated with a natural product, and of all of the natural products, it's one of the ones that has less data on it or less studies, right? And so maybe not as rigorous or not as effective. Another factor of this is for all ticks, right? And so perhaps Lone Star ticks or Longhorn ticks are influencing this data point as well. Other outline data that we have here. Um, these particular data are with pyrethroids, so we would expect them to be working much better, right, for their treatment. At least one of these yards, I spoke to the homeowner personally while we were sampling because she's like, did you find anything? I treated a little earlier this year to get a jump start on it. So she treated in April instead of treating in May. And so that treatment in April may not have had much of an effect on our sampling in May, as we would expect for a traditional treated property. And so these conversations are important when we're talking about data analysis and how we want to handle these things, and also with communication with homeowners, right? She thought she was getting a jump start to the summer season, when in reality, she may have missed that initial increasing tick population in May. One of the questions that we are curious about is, okay, this is for all ticks, but what if we just zoom in on black-legged nymphs, right? And so for black-legged nymphs, looking back again to 23 versus 24, our treated versus untreated, these averages are pretty, you notice the difference, right? The difference is very evident. But again, adding in 25 is still giving us a little bit more of a murky signal. If we break this up using the different types of treatment overall, we still have these two outliers here. The one that was green, that was way up high, is no longer here because that tells us that it was because of other tick species. So it's likely because of the longhorn ticks or the Lone Star ticks. We had a couple of sites where we just collected a ton this year. But for black-legged nymphs, we're still noticing, although the pyrethroids here are the highest, this could be due to that timing of treatment like we discussed. So in terms of Project Edge and tick treatment overall, tick population dynamics are changing, right? What we may have suggested to a homeowner 10 years ago may no longer be reflective of what would be effective in their backyard now. Yes, longhorn ticks do not bite people as frequently as others, right? But they still do. And they are biting pets as well. And so there's still a concern from a public health perspective. More so, Lone Star ticks are aggressive and they are also increasing in our area, and they also may need different recommendations for tick control than black-legged nymphs. And so as these tick population dynamics change, how we answer is tick control helping may change slightly as well. And then navigating with homeowners what is best versus what is acceptable to them, right? I may tell someone that a good bifenthrin spray that's well timed is best for getting rid of the most ticks in their backyard, right? But to them, they maybe don't touch my yard. I want to keep it as natural as possible, right? And finding that balance between how do we keep people safe and how do we keep the environment safe or their priorities protected. Stay tuned for Project Scratch. We have a new postdoc at URI, Emily Burton, and she's really dove directly into this. And so hopefully we'll have more to share on that, or she'll have more to share on that with Dr. Carrette. And so this, I think, is a big facet of what we'll do with its results and how we communicate this to homeowners. So overall, is tick control helping? We need to look at the timing of the treatment, the tick species that are present, and also one of the future directions, right, when I have more time to dive even deeper in the data, is I want to look at this combination of treatments. And so we have a few homeowners who use minimal risk natural products as a spray in their backyard, but then they also use tick tubes. And so it's kind of this hybrid between synthetic chemicals, right, and then natural products. And what does this look like for their results overall? We also had a variety of folks opt to apply pyrethroids themselves rather than hiring a PCO. And so all of these variables can be broken down and see if we can see any signal amongst all of the data so far. Stepping away from Project Itch for a moment, I'm going to do a quick aside. I wanted to do a shout out to Jane's newsletter that she's been sending us, right? The new VEC newsletter that has all of this great information in it. This month talks about a bunch of things. I know right in front and center is a great picture of Emily with a possum, so you can't miss that. But also, there is a behind the scenes look into what takes up most of our winter months, right? And it's this raising the tick colony. And this is one of the cores of our research for NUVAC. And so here are a bunch of fed black-legged tick larvae that I collected. So in the article, it talks about it's time to make ticks. We go out into the field this time of year to collect the adult black-legged ticks, so that way our colony stays as close to wild as possible. Those ticks are then fed on lab animals to allow them to engorge, and then they'll lay eggs. We have to make sure that all of this is controlled right and timed correctly. So that way, when the larvae do hatch, we have a clean colony as well as a pathogen infected colony for Borrelia burgdorferi, the causative agent of Lyme. And this full process takes a lot of patience and a lot of checking on ticks, not doing a whole lot of anything other than hanging out and waiting to molt. But it also takes a lot of work for animal coordination and making sure that we have proper IACUC protocols and that we're coordinating with agencies across campus, including the vets and the vet staff, to make all of this function, right? And make sure that animal welfare is taken care of, as well as maintaining our research standards. So it takes a really huge effort that I knew of before last year, but really began to understand last year being hands-on in this process. But by the end of all of this, we end up with about 15,000 to 20,000 ticks that are used for studies throughout NUVAC, from behavior, repellency, control. And by having this colony, it lets us run scientific experiments that are more controlled, as well as more rigorous overall, because we're removing a lot of the variables from just collecting ticks from the wild. And we're keeping all of the ticks one generation from wild, so that way we're not getting any sort of like lab discrepancies. When colonies are kept for too long in a lab, they note that they behave differently from wild tick populations. A quick snapshot into this. If my videos work, we will find out. One of the questions I get asked, at least from my friends and family, when I'm talking about collecting larvae from pans, is what? But the smaller animals that we feed ticks on are in these suspended caging, so that way it's on the bottom is mesh. And then with the animal on top, as the ticks feed, the ticks will fall through the mesh and they'll land in this pan full of water. As you can imagine, ticks are not the only thing that land in this pan of water. So there's poop and food and fur, lots of different things that we have to fish the ticks out from using a paintbrush. And so what you see here on the left side of the screen is that pan that was underneath the cage. And then you'll see me collect the ticks. Let me theory. Yeah, okay. They're time lapses. I promise I don't move this fast. But that's me collecting the ticks from the pan that's been underneath the animal, right, at the dirty stage, and into a pan of distilled water. And so that way the ticks kind of get rinsed off. We don't want them to collect. If you've ever kept ticks in captivity for any period of time, they get very moldy very easy. So by getting this first initial bath, that helps with that. Second from that, we take them from the pan of water onto a paper towel. So that's this piece. Things in life that are harder to do than you think they are is getting ticks out of water. And then from the paper towel, we rinse it again with DI water, combine them into clusters, and then store them in a vial afterwards. So that way we know exactly how many ticks are in each vial. Yeah, and emphasizing that this is a very, very light day. These are some ticks on one day from white-footed mouse. If we have a hamster, we can feed up to 1,000 ticks on it at a time. And so this looks very different, and the amount of time that it takes looks very different. So again, very, very light day of larvae. But yeah, just a little insight behind the scenes snapshot of what we do. And I just wanted to do a shout out to my colleagues here at URI who make this possible. Tom, our wonderful interns this summer, PhD student Domingo, and then Dr. Cred as well. And then, of course, the homeowners, they are fantastic. I've really developed great relationships and I love talking to them and seeing how invested they get into it. Some folks are not, and that's fine too. But a lot of folks want more answers and they want these answers, and they're really supportive of our science, and I greatly appreciate that. And also shout out to NUVAC for all their support and as well as all these discussions that really help make our science stronger. Questions?
Thank you so much, Lexi. That was awesome. I loved seeing those videos.
Yeah. I'll have to get one for my hamster this year, and that way you can like see the magnitude.
Yeah. Yeah, we can take questions. Anybody have questions? Lexi, can you see the chat or do you want me to read this?
Oh, okay. Chat.
Matt asks, can you describe more about the relationship between Project Itch and Scratch?
Yeah, so Project Itch is the sampling homeowners' backyards, navigating what tick control is working or not. And then Project Scratch gets more into this disease decision analysis and navigating how to get homeowners the best choices for them. So they put in their preferences, and then also we elicit experts to navigate what methods they would best recommend given to accomplish certain goals. And so meeting the homeowner's needs with what experts back based on what the homeowner wants to do creates this great tool. And so that's where Project Scratch comes in. And so um, that is now Emily's corner of the world with Dr. Corrette. And so we're really excited to see what comes from that. I know there's a lot of interest for it.
They will be doing a work in progress on
Yes.
that subject in December, I believe, as well.
I forgot to get the date. Thank you. I was like going to do a shout out.
It's December 3rd. Yeah.
Yeah, so species differences, what species differences did we see in 2025? We saw a lot of longhorn ticks and more Lone Star ticks. And so there are more ticks overall, but it was amazing to see how far the longhorn ticks have spread in our area so far. And the areas where they are good. I know recently our intern Mason there went out to one of the properties where we collected a lot of longhorn ticks this summer for Project Edge. And there's a ton of longhorn tick larvae. So they are here, and what that means for tick control, what that means from a public health perspective, I think we're still navigating.
Sorry, just to follow up.
Yeah.
Did you see any regional variation within the state in terms of the longhorns, or was it
Yeah.
really localized or?
So mostly southern, like so the
Mhm.
more southern and coastal areas are the areas where we were seeing longhorn ticks.
Okay.
Lone Star ticks, at one point, I remember Tom's talks early on, we're talking about, oh, Jamestown, like Eastern, and we're starting to see them kind of cross the bridge now. And so they're moving their way in from the east as well.
Wow,
Um,
interesting.
so just from last year, I know Mason's project for this fall is looking at longhorn ticks and how this has changed over time using the itch data. And so I'm excited to see more results on it. But just seeing them move from like pretty coastal and then starting to work their way inland a little bit is really interesting.
Yeah, it'd be interesting to see what happens here in Massachusetts as well. Good,
Yeah.
thank you.
Mhm. Hey, if you want to.
Was it my turn?
Yes, I was like, do you go by Will or Bill? I couldn't remember.
I'm Bill.
Okay.
Yeah.
Yeah.
Thanks, Alexis. I really enjoyed that update. I actually thought it was really cool to see how you fish those larvae out of water. That was awesome. I've actually done that a few times, and so I have a very specific question that's going to sound strange, but how often, when you're getting those larvae out of water, are they like moving around, or how often do they look lifeless and almost dead versus like moving? Or I know it's a very specific kind of question.
So in the water itself, they kind of look like they're on the lazy river, right? They kind of float around. But if they get to the edges of the pan, they will start to find their way out very slowly. And we change the pans often so we can catch them. but otherwise they just kind of float. But once they make contact with like the paper towel or something or the paintbrush, they will start to like diffuse out.
Well, let's put it this way. When you take them out of the water, do they wake up or do they look like they're motionless
Wake up.
very
I
quickly?
Yeah, I have done the underwater survivals where they take a minute though in a past life in grad school. Um,
I'll send you, and there's a reason I'm asking this, but it's not worth bringing up here, but I'm trying to interpret something that I saw, but I can just message you
yeah,
separately. I'll tell you what's going on. Thank you. I appreciate it.
yeah. Any other questions?
So, Bill, you might be interested. Back in the day when I had a hot tub, people always said, oh, I went swimming, so I probably washed all of the ticks off, or I sat in my hot tub and that probably killed all of the ticks. So I put them in those same vials with mesh tops and sunk them to the bottom. And not the fed ticks, but the unfed adult and nymphs. They actually remain alive after 2 days at 102-degree temperature hot tub water. So
Wow.
people, and what their thought is and what actually is often two different things.
Yeah, we're working on publishing Holly Gaff and I from Virginia, but we did a study with washing ticks, right? If you wash and dry ticks, but it was with Lone Star ticks. And so the heat, it's amazing how much heat they can survive with water. And one of the other studies looked at freshwater, brackish water, and salt water and how long Lone Star ticks could survive underneath. And it amazing. But we would call it zombie ticks because they would look dead and then essentially just like within the next 24 hours rewrite themselves and be able to crawl away.
Got a question about longhorn ticks. Are you finding larval clusters similar to what we see in the Lone Star ticks?
Yes, yep.
Is there any way to kind of distinguish the two besides getting them under a microscope?
Microscope's your best bet. They spread the same, they look the same, they collect the same. My weird thing is that they walk differently with their front legs. And that's something that I notice. I call it a monster walk, but they move more out and over. It's very nuanced. And the fact that I've stared at way too many between my time on Long Island, looking at ticks out on Montauk for way too long. But that is not like a key morphological difference. Other than that, microscope, I think, is your best bet. Unless, Tom, do you have a method?
I've got 70-year-old eyes now, so I have more and more trouble seeing these nuances. But I would say my experience about a week ago, we went to a place where we know we have both Lone Stars and Longhorn ticks, and within really 5 feet of the beginning of the trail, little sprigs of grass that have now have little seed heads, so they're like 5 inches above the ground. I reached down and everything that looked like a seed head had 20 to 100 larval ticks on them. And when I had them on my finger, I could tell that their mouth parts were shorter. And those are the longhorn ticks. But, that just shows the level of risk because, just imagine little sprigs of grass down a 100-meter trail. Probably little sprigs are about every 2 or 3 inches. And almost every one of them had a little grouping of larval ticks on it. Incredible, really.
Nita just put in the chat too about how you had previously described the longhorn tick larvae will like explode onto the flag. And I'd say like I see Lone Star tick. If you get enough Lone Star ticks, they do a very similar like it looks like diffusion, like the cliche physics class diffusion.
They both walk faster than black-legged ticks do, the larvae. So the larvae, black-legged ticks, stay a little bit more clumped on the flag as you're collecting them. And very quickly, the Lone Stars and the Longhorns will start to disperse.
Yes, and if anybody would like to help me quantify walking weird in a scientific way for the longhorn ticks, that would be helpful. It's an observational thing, but I've seen so many of them over time.
Yeah, I'll keep an eye out because I've
Yeah.
been collecting longhorn ticks for a couple years now in the vineyard, but I'm not sure if I've collected a cluster yet. Just adults and nymphs so far, but I was trying to see if I could avoid getting every cluster I collect because I get so many Lone Stars and double check under a microscope, but it seems like that'll be the best way until I get some practice and get a lot of catch a lot.
How do you collect your larvae? Do you use painter's tape or do you use
Lint
lint
rollers
spray? Yep.
directly on the flag, and I just do the best I can. I understand there's always going to be some embedded left in the fabric, but yep, just simply lint rollers. And I like to laminate them and use them as outreach tools.
Smart, smart. I like to hear how we all survive larvae differently. So that's my surveillance nerd, but thank you.
I would add that it's a sad day for me in Rhode Island because we used to only have black-legged ticks and occasionally American dog ticks. And now we have all of these other ticks that they just run amok. And so I've had to change the way I think about it all. And if the Lone Star ticks get any worse, I may have to move. I don't know how people live with them in the states to the south.
I was gonna say, I think I have this bottom photo here. These specks are all adult Lone Star ticks, and that was like maybe 5 paces down a trail when I worked for Suffolk County Health Department at one of the parks, and their Lone Star populations are crazy, but this was like a light flag. Like this wasn't a lot of ticks compared to some of the other locations where they are able to really like exploit the host systems and increase their populations.
I think it's an important fact that, New England didn't have to think about these for a long time, but now we are having to think about them. So it's just something that all of NUVAC needs to keep on their radar.
Any more questions? Well, if there's, oh,
Awesome. Thank you, Johanna.
yeah, I was just going to say, Johanna's going to do our next work in progress on the 22nd of October. So we've got Project Itch, Project Scratch, and she's going to talk about Project Listen. So can look forward to that. But thank you, Lexi, and thank you everyone for joining us today.
Thank you all.